Hello, good day everyone. We are from Group 4. We would like to present our topic, eukaryotic microorganism and parasites. So our learning objective, by the end of this lesson, students should be able to describe the characteristic of various eukaryotic organisms, explain the function of organelles found in a eukaryotic cell, describe the morphological characteristic of protozoa, helminths, and fungi, list examples of pathogenic eukaryotes and the disease that can be caused. Okay, first we go through the term eukaryotic. It's originated from the Greek words. Eu means true and karyo means nucleus. So it literally means true nucleus, which means this eukaryotic organism has true nucleus. And the characteristic, it undergoes cell division called mitosis and meiosis. And the genetic material for this organism is located in the nucleus with a well-developed nucleolus. It's mostly multicellular by nature, but some eukaryotic organism is unicellular too. This organism has membrane-bound organelles, either single or double membrane system. It has no plasmid except for mitochondria and chloroplast. Is autonomous with their genetic material. Eukaryotic has cytoplasm and cytoskeleton. The DNA is wrapped around histone proteins, and the chromosome has linear DNA compared to the circular DNA in the prokaryotes. So this is the difference between plant cell and animal cell in eukaryotic cell. For the plant cell, it have cell walls made of cellulose. It also has plasmodesmata, in which it allows communication between cell. It have large bulk wall, and chloroplast to allowing photosynthesis. Meanwhile, the animal cell, it contains peroxisomes and lysosome. This one lah, based on the picture. It also has centrioles to form the spindle fiber used during the cell division. Eukaryotic organism includes protists, fungi, plant, and animal. So this is the example lah, unicellular, like protozoa, for fungi and algae. It either unicellular or multicellular. Helminth, it's in the multicellular type. For the external characteristic of eukaryotic cell, external structure of eukaryotic cell, it have locomotor appendages. It's either it's cilia and flagella. Eukaryotic flagella are thicker than prokaryotic flagella. It's more complex, covered by the extension of cell membrane. And the flagellum contains microtubules, and the cilia shorter and more in number. For the surface structure, eukaryotic cell have glycocalyx, in which it composed of polysaccharides, fiber, network, and slime. Similar to the capsule of prokaryotes, the glycocalyx important for protection, cell adherence, and signal reception. Its location, it's beneath the glycocalyx. For the fungi and algae, the surface structure is thick and rigid cell wall, and protozoa, some algae, and animal cell do have cell membrane. For the cell wall, for fungi, they have thick inner layer of polysaccharide fibers composed of chitin or cellulose with thin outer layer of mixed glycan. And for algae, it's commonly found cell wall component as cellulose, pectin, mannans, and mineral. And for the membrane, it consists of phospholipid bilayer and sometimes contains sterol. Internal structure of eukaryotic cell. This is the picture of the nucleus. The nuclear matrix is known as nucleoplasm. And nucleolus is condensed with the RNA content. Nucleolus is the main site for ribosomal RNA synthesis lah. And also collection area for ribosomal subunit, which are then transported to the cytoplasm through the nucleopores. And this chromatin location in nucleus, it contains chromosome and carries the genetic information in the cell. Okay, this is one of the organelle found in eukaryotic cell. Endoplasmic reticulum, ER, is a series of membranes distributed inside the cytoplasm. Consists of broad, flattened, interconnecting sac and tubules. It also have this called cisternia. The interspace of the sacs and tubules is divided either into the rough endoplasmic reticulum with the ribosomes attached. This is the place where proteins are synthesized and modified to be secreted out of the cell. And for the smooth endoplasmic reticulum, no ribosome attached. The function is to produce lipids, phospholipids, cholesterol, and steroid hormone. Okay, next organelle of the eukaryotic cell, Golgi apparatus. Golgi apparatus is a flattened membranous sac with cisternae stack arrangement. The function is to modify specs and distributes proteins and lipid manufactured by the rough endoplasmic reticulum and the smooth endoplasmic reticulum. So this is how the protein produced in the rough endoplasmic reticulum. Firstly, the protein produced at the ribosome of rough endoplasmic reticulum are transferred into the cisterna. It will form a transport vesicle from the ER membrane. Then transport vesicle move from the ER to the GF fuses with its membrane, releasing the protein into the cisterna. Then the Golgi apparatus concentrates and modified the protein into glycoprotein or the lipoprotein. Then the secretory vesicle are formed using the membrane of Golgi apparatus. The secretory vesicle transport the protein to the plasma membrane, and then protein secreted out from the cell through the process called exocytosis. Okay, lysosome. One of the organelle too is the membrane-bound vesicle that pinch off from the Golgi apparatus. It's contain variety of hydrolytic enzyme. The function is as the intracellular digestive system to digest nucleic acid, proteins, polysaccharides, and lipids. It also digests non-function organelles known as autophagia. For the mitochondria structure, okay, this is the picture of mitochondria structure. It is a major site for ATP production. ATP is the energy production for the cell. It's the mitochondria is responsible for oxidative metabolism and most ATP synthesis. It's have crystia here, cristae. The infoldings found in the inner membrane, while the matrix is carry the enzyme needed for Krebs cycle, DNA, and ribosomes. The inner membrane of mitochondria embeds the enzymes used for the electron transport system. There are similarity between the mitochondrial structure with the structure of a single bacterial cell. Mitochondria carries their own set of DNA, enzyme, and ribosomes. For the eukaryotic mitochondria contains prokaryotic ribosomal material. Okay. Chloroplast. Chloroplast is found in algae and plant cells. It don't have in animal cell lah. Photosynthesis. It's go through photosynthesis. It's the function for chlorophyll is to go through photosynthesis, which it converts energy from the sunlight into chemical energy. The thylakoids are stacked on the top of each other, forming a grana. The chloroplast also carries chlorophyll and other pigment.
Good evening, everyone. My name is Mastasha Ramli. I'm going to present my slides about protozoa. So here, protozoa is a free-living organism, and it found in almost every possible habitat. So the person who found protozoa was Anton Liu Wenhoek, who's the first person to see and found protozoa. And he described protozoa as a free-living things. And it's also for several parasitic species. It's from animals, and Giardia lamblia from his own stools. Okay, next. The general characteristic for protozoa. Protozoa is from a Protista kingdom, and is also a eukaryotic cells that has DNA and well-defined chromosomes. Protozoa also a unicellular and multicellular, and it also have asexual and sexual reproductions, like binary fissions and multiple asexual divisions. In some types, it can be both sexual and asexual reproduction and takes place. Mostly protozoa are aerobic, but some are anaerobic. For example, intestinal protozoa. Protozoa are some free livings, and some shows paternalistic effects. There are also some autotrophics. Protozoa is also a heterotroph, where they have like food consumption through phagocytosis. Sorry. Okay. Next slide. For the shape, protozoa have a dynamic shape and sizes, and it also shows diversity in their shapes because they lack of cell walls. So the shape determines by their flexibility and rigidity of their ecoplasms right beneath the membranes. So protozoa generally enclosed by a plasma membrane only. If more than one layer of plasma membrane is involved, it's known as pelecyl. Sorry. Okay, so in parasitic form, protozoa can have a thick protein surface coat, such as like glycocalyx, give the protozoa immunological advantage. Okay, next. So this shows the carry complex organelles in their cytoplasm or endoplasm. Okay, the first one, mitochondria. It carries an enzyme for oxidative phosphorylation TCA cycle. For Golgi apparatus, it has a different roles in different protozoa. in amoeba as a source of skeletal plates, microsporidians, parasite as a polar filaments. For microbodies, it contains enzymes. Name differs based on enzyme that they carry. For example, like peroxisomes and hydrogenisome. Next slide. This is the classification based on the protozoa locomotions. For the first one is Mostigophora. For the movement, it's moved like a flagella. And for the reproductions, it's a binary fission longitudinally. For Sarcodina, the movement is pseudopodia and the production is binary fissions. Next, for the Sporozoa, the movement is often non-motile, but sometimes with flagellae, sometimes it's creeping. And for the productions, it has a multiple fissions. And the last one is Coleopora. The movement is cilia and the reproduction is binary fissions and transversely. Okay, next is Flagella. So it's a singular that called flagellum. Flagella are web-like extension of the cell membranes consisting of an inner core of microtubes arranged in a specific configurations. So two single central microtubes surrounds by 9 principal lobes. Okay, next is cilia. Cilia, for the singular, it's called like cilium. It indulges in wave movement, allowing cells to swim in fluids. It's similar in construction to flagella, but with the interconnecting basal elements, facilitating synchronous movement. Next is protozoan parasites. So most humans parasites go through 3 general stages. The first stage is microbes transmitted to human through soil, water, food, animals, and humans. The second one is microbes invade and multiples in the host, further producing more parasites to infect other hosts. And the last one is microbes leave their host after multiplications and enter new hosts, multiple hosts involved. Some parasite needs vector to carry the disease. And for the photogenic protozoa, for example, like Trypanosoma brucei cause sleeping sickness and Tzitzit cause Segas disease. Both parasites occur in the blood during infections stage, transmit by blood-sucking vectors. So this is the image of Trypanosoma brucei and Trypanosoma brucei in thin human blood smears. For trichomoniasis, parasitic urogenital disease caused by Trichomonas vaginalis, it's in fact urogenital tract surface in both males and females, and it's also feed on bacteria and cell secretions. Disease transmissions, the first one is sexual intercourse, and the second one is children can be infected through contaminated linens and toilet seats. And the third one, it can be transmitted by sharing towels, sheets, and undergarments. It's in fact the vagina only when the vaginal secretion have abnormal pH. For example, like optimum pH for the organism is 5.5 to 6.0. For the symptoms, the first one is intense itchings. The second one is copious white discharge in females, raw egg white consistency, and the last one for males, there are asymptomatics. Next one is the diagnosis for micro speaks examinations of venereal smears and uterus secretions. And the treatment is metronidazole flagyl. Flagyl used during pregnancy can cause abortions. And the third one, it's important to prevent infections before delivery to prevent infections of the infants. And the next one, it's restoring the normal vaginal pH in women. And the last one, it's vinegar douche is effective. Trichomonas infections can cause early rupture of membranes and premature damage. Okay, that's all for me. I'm going to continue with my next group mates.
Okay, good evening, everyone. I'm Annie. I want to present next slide for our topic. This is a helminth, another organism. It divided to two class, phylum Platymintes, phylum Nemathamintes. It's also divided to two class, Cestodia and Trimaldia. And then phylum Nemathamintes is one class only, Nematodia. Okay, next. Okay, general characteristic of helminths. It is not specific organ for locomotion, moved by muscle contraction in erection, process primitive nervous and accessory system. The integument cuticle anomaly arm to resist internal digestion, the evolution of alimentary tract. We develop reproductive system. Okay, next. Another organism is chestnut, also known as tapeworm. Body structure like taper-like and segmented. Both sex is a hermaphrodite, both male and female characteristic. Head possess hook in sucker, no alimentary canal, and no body cavity. Next. Pathogenic chestnut. It's a Taenia solium. It also called pork tapeworm. It definitive host human, intermediate host pig, transmitted through human feces and contaminated fodder. Next, Taenia sagineta. My God. It's also called it's a beef tapeworm. A definitive host, human, can be human and harboring the adult worm. Intermediate host, cattle, infect human who eats raw meat infected with larva. Dipilobotrium latum, infect human when raw uncooked fish infected with cyst is eaten and causes intestinal infection in anemia. Next. Another organism, trematode, it also known as a flax, body structure not segmented, leaf-shaped body, sex mostly hermaphrodite, both male and female characteristic, head no hooks to sucker, incomplete alimentary canal, and no anus. No, it's also no body cavity. It's a flexible lah. Okay, next. Another organism, pathogenic trematode. It is Schistosoma mansoni black flax. Infect human when the, how to say, Sal?
Cercerai.
Cercerai is released into the water through infected snail. Mature in the human liver and lung, travel through the bloodstream. It is causes schistosomiasis. The symptom is migratory pest, is dermatitis. During acute pest, Katayama fever, and a chronic phase, intestinal schistosomiasis. Next. Wait, Okay, next organism is nematode. It also known as a roundworm. Body structure is cylindrical and unsymmetrical. Sex can be male and female. Head no hook and suckers. Head buccal capsule. Complete alimentary canal present and body cavity present. Pathogenic nematode Ascaris lumbricoides, largest nematodes, adult female 25 cm and adult female can be 30 cm. Infection through soil contaminated with human fecal matter. The ova can remain infectious for years, resistant to chemical and temperature. Causes ascariasis can attack the human lung and intestine. The symptom is coughing, gagging, abdominal pain, vomiting, and weight loss. Major complication is intestinal and duct blockage, poor nutrient absorption leading to stunt growth in kids. Okay. Next is river blindness, how to pronounce?
Onchocerciasis.
Onchocerciasis.
I think so.
It's caused by larva of Onchocercia volvulus, roundworm, adult worm, microfilariae accumulate in the skin nodule. Transmission is by vector, black fly bites infected individual, ingest microfilariae in the mixture in the fly and move to its mouth parts. Infectious by microfilariae enter the new host skin and invades tissue including the eyes. Adult worms causes skin nodule and abscess. Microfilariae causes skin depigmentation in severe dermatitis. Diagnosis, microfilariae detection in the skin sample, visible adult worm through the skin. Treatment, usually treated by ivermectin kill adult worm and microfilariae quickly. Prevention, eliminate the vector and backfly. This is the picture blindness caused by onchocerciasis.
Hi, good evening to all. I'm Siti Aisha. I would to present about loiasis. Loiasis caused by lau eyeworm transmitted to human by deer flies. Adult worms live in subcutaneous tissues and eyes. Microfilariae appear in peripheral blood in the daytime. Microfilariae concentrate in the lungs at night. Worms develop in the flies, migrate to their mouthparts, and transfer to other humans when the flies feed again. The microfilariae migrate through the subcutaneous tissue, leaves trail of inflammation, and settles in the cornea and conjunctiva. Diagnosis: finding microfilariae or worms in the skin or eyes. Treatment can removing adult worms and ceramine to eradicate microfilariae. Control: eradicating deer flies, like this picture. And next is lung fluke. Lung fluke cause is Prognomus wastemani. The disease transmission causes infect human when shellfish infected with the metasaccharida of the fluke is eaten. When the metasaccharida will leave the crab or crayfish when it is digested in the human small intestine. The larva will bore through the intestine and in the abdominal wall. Next slide is the larva will leave the abdominal wall and penetrate into the diaphragm and the membranes around the lungs to reach the bronchioles. The will produce adult will lay eggs in bronchioles, and when the human cough, egg move to the pharynx and swallowed and exit through the face. The symptoms is disease is chronic cough, bloody sputum, breathing difficulties. The diagnosis is X in sputum. Treatment like a praziquantel, cooking shellfish before eating as a prevent to avoid the disease. This is we reach in conclusion for topic today is general summary or conclusion is eukaryotic cells are large and complex cells containing membrane-bound components called organelles. And major organelles is in an eukaryotic cell are the nucleus, mitochondria, chloroplast, endoplasmic reticulum, Golgi apparatus, and locomotor appendages. While fungi and protista are the major kingdoms that contain eukaryotic microorganisms. Type of disease due to pathogen is tapeworm infection, cause Taenia solium or pork tapeworm and Taenia saginata or beef tapeworm. Infection by eating undercooked or uncooked pork or beef, and through contact with while contact through with dogs and eating raw fish. Disease transmission can enter to body as larva in raw meat, especially pork. Adult tapeworm develop into the intestine, absorbing large nutrient quantities, leading to malnutrition. Can cause malnutrition. When human ingest or auto-infected by tapeworm eggs, the released larva can penetrate the intestinal wall, entering the bloodstream, and the larva can develop cystic circus or bladder worm. Okay, bladder worm can have oval white sac with the tapeworm head invaginated to it. Then in human, calcification fails to stop the growth of Certisaki. Certisaki patient can suffer paralysis and convulsion. When the Certisaki dies, toxin is released, causing severe or fatal allergic response lah. Sanitary disposal of human waste through cooking of meats and fish to prevent this disease lah. This is a picture life cycle of Taenia, and then the Philobothrium latum is what we call another broad fish tapeworm lah. Infects women through ingestion of raw or poorly cooked contaminated fish in sushi and other dishes lah. The worm coil up in the fish muscle will reach and mature in the intestine. Adult worm then attach to the intestine producing eggs. Next slide is this worm needs small crustacean and fish as intermediate host to complete the life cycle. This parasite absorb large amount of vitamin B12 causing anemia lah. Diagnosis eggs or proglottids in the feces, and treatment doctor can give niclosamide and anti-helminthic agent to prevent this disease, avoiding raw meats and fish or infected dogs. This is a life cycle of the Philobothrium latum. This picture show you the broad tapeworm and bladder worms. So thank you for your attention. I will leave it to other friends to continue the presentation.
Good morning, everyone. I'm Nelly. Now I will to present our question of the statement. Explain the component of phospholipid bilayer of plasma membrane. Phospholipid is the basic structural element of cell membrane. Phospholipid serve as a partition separating the interior and exterior of the cell. A hydrophobic barrier is produced by the structure of bilayer, which has hydrophilic heads on the outside and hydrophobic tails facing one another. This is hydrophobic head. The head group, typically a phosphate containing molecule, is attracted to water and therefore face the aqueous environment inside and outside the cell. And this is the hydrophobic tails. The tails, composed of two fatty acid chains, are not soluble in water and are therefore buried in the interior of the bilayer. And the next question is function of plasma membrane. The plasma membrane is a fundamental component of a living cell, playing a vital role in maintaining cellular integrity and function. The importance and function of the plasma membrane of the cells include selective permeability, cell communication, structural support, cell recognition, and transport of molecules. The selective permeability, the plasma membrane controls what enter and exit the cell, allow essential nutrients and oxygen to enter while removing waste, act as a protective barrier. As a cell communication, contain receptor for signaling molecule as a hormone, enables cells to respond to environment changes and critical for intercellular signaling in tissue. As a structural support, maintain the shape and integrity of the cells, anchor the cytoskeleton and extracellular matrix, essential for the cell stability and movement. And as a cell regulation, contain glycoprotein and glycolipids for identification, help the immune system differentiate between self and foreign cell, vital for tissue formation and immune response. As a transport of molecules, facilitate active and passive transportation, use protein channels and pumps for movement, balance ions and molecules inside the outside the cell. And the conclusion, the plasma membrane is essential for cellular survival and functionality. It's not only protect the cell, but also facilitate interaction with the environment, ensuring homeostasis, communication, and coordinate activity with multicellular organisms.
Hello, good evening. I am Mefoni. Then I will present the last question, which is the third question. For this question, I'm sorry, I deliberately use Microsoft Word to expand so that we can see everything in once. So for the third question, actually the question is about prepare a detailed write-up on the various membrane transport mechanism in the cell. Then please refer on the table. So there are 7 various membrane transport, which is number 1 is diffusion transport mechanism, and the description actually it was a random movement from high to the low, and the substance transportation it use a lipid soluble molecule can diffuse through the lipid bilayer. So the example of the diffusion is oxygen, carbon dioxide, the urea creatinine. So the transport mechanism osmosis for the description is a passive movement of water across a semi-permeable membrane and from high water potential to the low water potential. So substance transportation is a water molecule and transport only water across membrane. Same to the description is from low solute high water to the high solute low water. So the example for osmosis is a water, plant cell, and red blood cell. Number 3, transport mechanism. This is a filtration. It's a passive process of moving material through a cell membrane. It's a passive energy, high static pressure occur across semi-permeable membranes and from high pressure to the low pressure. So substance transportation is a water and soluble molecule. It only can pass through using a small molecule and substance across membrane due to pressure gradients. So the example for this filtration is a water, urea, amino acid. So next is a facilitate diffusion. Same as filtration, passive transport where molecule move across a cell membrane with help of transmembrane protein without energy is a what we call it as a ATP from high to low concentration. For substance transportation is a membrane protein molecule able to across the lipid bilateral to pass through the membrane. So the example is like glucose transport or ion channel. So next is number 5. The answer for active transport. So description is a cellular process in which substance move against their concentration gradients is from low to high concentration using energy. So there are 2 types, which is primary and secondary active transport. Then the substance transportation is a moving materials against their concentration gradients and using energy from cellular respiration. And for the types, we have a primary and secondary. So primary active transport, they directly use ATP hydrolysis to move substance across the membrane. And secondary active transport use the energy stored in the electrochemical gradient of one substance created by primary active transport to transport another substance across the membrane. So the example for the active transport is as potassium, sodium, calcium, and amino acid. Then we go to number 6, endocytosis. Description for this is a process by which cells take in substance from outside of the cell by engulfing them in a vesicle. So it's a an active transport process and required energy lah, which is we call it ATP. So it's a wide range of substance into cell, macromolecules, particle, lead substance, and other cell. So substance transport, we have three, which is number one, phagocytosis. This type of endocytosis engulf large particle. So for pinocytosis, they are uptake of extracellular fluid and dissolved solutes. So for number three, receptor-mediated endocytosis, the target of specific molecule that bind the receptor on the cell surface, which is allowing for efficient uptake of particle substance. So the example for endocytosis is like micromolecules protein. Then we have a toxin, specific ligands, substance like cholesterol. We can see via LDL. Then number 7, exocytosis. So the description is the process of taking a substance from or particle from inside of the cell and transferring it to outside the cell using a vesicle. Then the substance transportation is a crucial transport mechanism that enable polar molecules to flow through the cell membrane, so hydrophobic lipid bilayer. Newly made membrane protein and membrane lipid are moved on the top of plasma membrane. So for this the example is like hormone, insulin, and glucagon. So in protein, some essential protein. Then this topic of the question have been presented. So that is the end of our topic today. Thank you.